Welcome back to another video, year 12s. Alrighty, so we're looking at natural killer cells for our recap. What are they? There are cells that like to destroy our own cells if they've been infected or damaged. And essentially they make the cell go into apoptosis. We have eosinophils as well. Okay, and again, they're granulated cells as well, just like the rest of them. They look a little bit different in their anatomy here. All right, but what they do is they destroy invading pathogens that are multicellular. So, anything that gets in, for example, tapeworms, which is a parasite, a large multicellular parasite, these eosinophils will attack that. Okay, not as good as at destroying little things, but when it comes to big things, they have DNases, RNases, proteases that help to break down these things. We also have interferons, which literally do what they're named after. They interfere with cellular processes. Okay, so they are a cytokine, also known as a signaling molecule, released in the immune system. And they react with the MHC1 markers on cells. Okay, so again, they see a cell that has been infected, as you can see here. And what they do is they interfere with the cells adjacent to those infected cells. So they don't actually do anything with the infected cell here, but they'll work to reduce the protein synthesis capacity of the adjacent cells. Okay, so what does that mean? They essentially come on in, all right, so they boom, and they reduce the effectiveness of this cell making proteins. And therefore, when this cell gets infected by these viruses, okay, it's not going to produce as many viruses. So yeah, these ones are pretty cool. Exactly the name. Interferons interfere with neighboring cells. And lastly, complement proteins. Okay, these ones are important too. So they are a part of the immune system, but they're not cells, they're proteins. Okay, and they just float around the bloodstream, as you can see here. Good old liver, literally keeping you alive. They're also made in the liver here. What do? They act as a chemoattractant, so they bring phagocytes to the area with chemicals, chemical attractor. They opsonize the bacteria, all right, they flag them for death. They essentially stick on and go, hey, phagocyte, engulf this. And they also may do a membrane attack complex, which they just destroy the cell membrane of the bacterium here. Okay, but really the biggest one here is that they are a chemoattractant. All right, as well as opsonizing protein to float around the bloodstream. So they don't go through the lymph, in the bloodstream only. All right, second line of defense, innate immunity, inflammatory response, and fever. So this one's a bit easier here today. So what is the process of inflammation? Essentially, if you look at your hand, we all have a part of our body that has inflammation at some point. If you look at somewhere you've been cut, it might be a bit red. Yeah. All right. Or somewhere where the skin's been broken, so it's no longer intact skin. That's where inflammation will occur. Okay. So there's a few different steps. Again, it's not that important to know these, but if you do know these, it will help you with the inflammation response. So initiation, vasodilation, and migration. All right, so initiation is when, the skin is actually split here. So in this case, it's a splinter or, something stabbing. It's pierced through the skin, and then the pathogens get in. So your little bacteria here are now in. Okay, so what's the first thing that's going to happen? The destroyed cells, or the cells around the destroyed cells, will release cytokines. Okay. Additionally with that, mast cells will break down. Remember, mast cells are all over your body, and they release this histamine. So they'll release it into a localized area. Okay, so say this is on your finger, and you've accidentally touched a thorn on a rose bush or something, and you've been pierced. All right, the mast cells will go to work. The cytokines will be released. That is the first step. What occurs next? Vasodilation. Okay, so if anything is a dilator, it increases it. All right. Vaso just meaning blood vessels. So histamine released from the mast cells travels to nearby blood vessels. All right, as you can see, it does a few things to the blood vessel. This is actually increased in size. All right, so this is now wider than what it was. All right, as you can see, there's more holes in the wall, so it's increased the permeability of the blood vessel. All right, there's going to be a lot of redness and warmth around the area as well, because there's a lot of blood there. Okay, awesome stuff. And that's because of that histamine. Then migration, all right, is a migratory pattern, so migration of your phagocytes essentially, and white blood cell leukocytes. So more blood means more immune cells at the infection site. Phagocytes are guided by the cytokines, all right, as they opsonize, secreted by damaged cells, and they find these pathogens and hopefully do phagocytosis. Complement proteins are attracted to the pathogens and make it easier for the phagocytes to destroy them as well. All right. And yeah, these phagocytes will move in and they'll do what they do best. Okay. They will engulf the pathogens here. Okay. Again, we should be aware that stuff like bacteria will replicate quickly. So sometimes your white blood cells can't engulf them fast enough. And that's why you get an infection or something. So that was the inflammation response. We also get fever that happens as well when we're infected by a pathogen. So the fever is a temporary increase in body temperature. So generally this is when you get some sort of, influenza, so the flu, or some viral infection, but it can happen in other infections like bacterial infections too. All right, a complex series of responses that raise the set temperature point of your body. Okay. And essentially that's to help destroy the pathogens that this little kid has, all right, or you have. So pathogens cannot withstand a high temperature and the enzymes will denature and break down, which essentially kills them. And that's essentially because they have an optimal temperature range, and if it goes out of that range, which your body can do, if you're 37 degrees, a bacterium infects you, the optimal temperature is probably like 36 to 38, and there you go. You get to 39 degrees, they will die.